talking about aspects of cells and organs of the immune system. And so like I told you last time, this is kind of our cast of characters for what we're going to be seeing for the rest of the semester. Another way to kind of think about this is that basically right now we are taking this sort of whole mess of all sorts of things that we see in the immune system, and we're going to try to break it into some groups, and we're going to try to organize it in some way so that we see what's what. The first way that immunologists like to organize all of this stuff that's in the immune system is that we like to talk about layers of immunity. And so we divide the immune system into a few different layers. In different places, sometimes you'll see something other than 3 layers, but I'm going to go with sort of the classic 3 layers. I've got a view from actually my virology textbook here and a view from my micro textbook on the right, because I think they both kind of get at a few different interesting things. And so you can see these different layers first on the left. We've got these 3 layers of barrier defenses or barrier immunity, innate defenses or innate immunity, adaptive defenses or adaptive immunity. And so one of the ways that immunologists divide up all of the different parts of the immune system is they say, okay, is this barrier, is this innate, is this adaptive? Some of you may have heard the term acquired immunity or the acquired immune system before. If you have heard that, that is a super out-of-date term. Most immunologists actually really kind of got rid of that term in like 2000. But it still persists in a few different places. If you've heard acquired immunity, that is adaptive immunity. So it has been renamed as adaptive immunity. I'll say a little bit about why that is a bit later, and a lot more of that in later classes. So we've got these 3 layers. Again, one thing to just be aware of is that these are 3 layers that we sort of made up as a way to help us understand and divide up the immune system. There are a number of cells and molecules that don't totally behave. I was actually here over the summer talking to a former student of mine who was teaching a thing over the summer, and we were actually having an argument over whether certain things were barrier or innate. And we were like, and I'm like, well, I consider them barrier. He said, well, I consider them innate, and we were going back and forth. So it's not like that every line is perfect in all of this, and we'll see that in a few places. But it is really helpful as a framework for us to think about. I, for a lot of time, have always used this version that's on the left to talk about the layers of immunity. One of the micro textbooks uses the version on the right, and in a lot of ways I do think it is a lot better. Just because sometimes in the one on the left, it makes it look like totally like they don't interact. They're 3 different walls that are completely different from one another. And in the right, they're kind of interacting a little bit more in terms of different ways of defending like this castle. So it's not like they're totally separate. It's not like they might not happen sometimes at the same time, or they might not interact with one another. So you can see that this castle has a moat, and it has alligators, and it has archers who have arrows, and there's like people pouring boiling oil, and there's like a wall, and like there's a bunch of different things. And I could, if I really wanted to stretch an analogy, tell you like why different, this one is like a moat, and this one is like an alligator, and this one is like an arrow, and things like that. And there are reasons why some of that's similar or different, but what you can notice is that some of these things are going to be like really specific, some of them are going to be fast, some of them are going to be slow, some of them are like, you know, actual parts of the structure of the castle, and some of them are more like the boiling, like a chemical is like, there's a lot of interactions between them, and that sort of level of interaction between all of these different types of defenses is probably more realistic than these sort of walls. So generally, when we as immunologists think about barrier immunity, we're really thinking about different sort of anatomical structures or sort of unique aspects of anatomy that try to keep microbes out, or that block microbes in general. They are not specific to any sort of microbe. They are super general, and the idea is really just keep the microbes out. And one of the reasons why, of course, that is important is that we talked about how quickly microbes reproduce last time, and this sort of problem of timing and reproduction. And so you want to make sure that you are sort of keeping the microbes out as much as possible, or making it an inhospitable environment for them to grow really quickly. So the biggest barrier organ that we have, of course, is our skin. Many people don't think of the skin as an immune organ, but it is actually keeping microbes out. And if you were to go to the hospital and see burn patients, the thing that burn patients usually actually have problems with is infections, because they have lost the barrier keeping microbes out. We can also think about things like the fact that your stomach has acid in it, so that if you ingest something, the acid might kill it. Or the fact that your lungs have cilia that are sort of beating like this, trying to expel things. Or other things like that. And so you can see there are things in like the skin, things in the GI tract, throughout the alimentary canal, in the airway and lungs, in the urogenital tract, in the salivary and lacrimal glands, which are tear ducts. All of those things that are basically just sort of barriers trying to keep the microbes out. In general, all of those barrier organs partially are working as actual physical barriers. So like they have unique types of junctions between the cells, so microbes can't sneak in. They have like a legit barrier where we're not going to have microbes coming in. So we've got sort of these physical barriers, that physical movement of the cilia, all of that kind of stuff. We also have some interesting sort of chemical barriers, like things like that pH I mentioned in the stomach. I mentioned that I was debating with a former student about that line between barrier and innate. Some of these chemicals I think of as innate, and he thinks of as barrier, so like that's where we were discussing. So like the barrier organs have a lot of innate immune defenses, is what I would say. Sort of like they're interlinked. More and more often, we're also actually realizing that some microbes who we have co-evolved with also seem to be part of this barrier defense. Your skin right now is actually covered with a layer of microbes that are nice, healthy friend microbes. And that means that there's no niche space for the bad guys. Your GI tract is full. Your lungs are coated. And so you've got all of these parts of your body that are basically coated by commensal organisms that make it so it's harder for other organisms to colonize. And so that's not even like part of your body, but technically that might be part of your barrier defense. And that is a big thing that people end up thinking about when they are, say, on antibiotics. But then the things that, to be honest with you, most immunologists don't spend a ton of time thinking about barrier defenses. We're not going to talk a ton about barrier defenses this semester. We're really going to be spending a lot of our time thinking about the other two layers, the innate and the adaptive layer. If you look at the course schedule, you'll see that Friday and next week, we're talking specifically about a lot of details of some aspects of the innate immune response. And so today I'm kind of giving you like broad definitions, a little bit of like compare and contrast between the innate and adaptive immune system. Don't worry, there is so much more detail that I could say. It's just not necessarily a today thing. But so don't feel like, if you're like, oh, but there's things I don't quite get from what we're doing right now, it's not like you have to know all of it today. But we do want to think a little bit about some of the compare and contrast of innate versus adaptive immune responses. And so there are versions of a table like this, pretty much wherever you think about immunology, sort of comparing and contrasting innate and adaptive immunity. One of the really key things about innate versus adaptive immunity is the difference in timing of when we really see their action against pathogens. So they both maybe are doing stuff a lot of the time, but kind of like the big splashy like thing you think about, thing you write home about, thing that you really measure, thing that you're excited about happens at different times. Innate immune responses are really happening within hours of you encountering that microbe. I often think of innate immune responses as being kind of the game you got for the first 96 hours after you're encountering that microbe. While adaptive immune responses, you can see here it says, are slower in response. It's more like they just take a while to get going. They start doing something early, but they're not actually really useful for a while. And so it's days to weeks before you get a really good, strong adaptive immune response. I would say that a peak adaptive immune response, I often think about as being somewhere between like 10-ish days, like 7 to 14 days, as being sort of key in a lot of cases. And that's awesome. Adaptive immune responses are awesome. But I want you to think about that timing issue for a second. And remember what I told you last time about the speed with which microbes reproduce. If you had to wait 10 days to have a response to any microbe that came into your body, you would lose every time. Because that microbe is going to be able to reproduce so quickly in those 10 days that it's going to take all your resources, it's going to take all your everythings. And so what you can imagine is that having a response that's really fast, having a response that's happening right away is really, 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 really important. I say that, and I'll show you a little bit about this on the next slide, and then I'm going to come back to the table. Because when I was in graduate school, in particular when I was an undergrad taking immunology, everyone talked about innate immunity as like super lame and boring. And you talk about it for like this much of the time in an immunology class, and the rest of the time is all about adaptive. Adaptive is the one with all the bells and whistles and fancy and everyone gets excited about. And everyone's kind of, if you didn't have the innate immune system protecting you while the adaptive immune system was getting revved up, you would die every time. Because you need something actually to work during that early period of time while you're waiting around for the adaptive immune system. And so that timing difference is one big difference. Oh, this wasn't exactly the slide I thought it was, but that's okay. I'll go around in the order I have. So what you can notice here is in this part of the graph, you can see this light blue line, which is showing us an innate immune response. The darker blue graph is showing us the adaptive immune response. And you see there is this period of time where the innate immune system is kind of all that's kicked on, or all that's doing much. Not until later where you're going to start to see this adaptive response. Again, I will come back to talk about other features of this plot, because this is not the plot I thought was right here. I thought this was right here. This was the slide I thought I was going to. And so this is sort of my, if anything, this is my argument to past me, that I should care about the innate immune system. I kind of do research on the innate immune system now, so I do care about it, and a bunch of people won the Nobel Prize for it recently, so like pretty important. But this is an argument I would make to my past self. First, we can look at this yellow line on this graph. This is showing the amount of some microbe on the y-axis and time on the x-axis. And this is sort of what happens if you get infected with something. Well, at the beginning, you don't have it. Duh. Then you get infected, and the thing starts to reproduce in you, and eventually, it gets stopped. We stop it at some maximum rate or maximum level, and then we get rid of it. And that's how it works. There are people who are born without adaptive immune responses. So there are some conditions where people are born without adaptive immune systems. The little boy on the right is one example of this. He has a disease where he does not have an adaptive immune system. Most of the people with this disorder are boys because this is an excellent trait. A lot of the genes that are involved in this are excellent. And this is a pretty severe disease. Most of these boys actually don't make it to age 20 without pretty serious treatment. And even with that, this disease is usually referred to as boy in a bubble syndrome because you have to live in a special environment entirely away from microbes. And so he's actually, you can't totally see it here, in an entirely enclosed environment, in a bubble, to be kept from microbes. So not having an adaptive immune system, pretty seriously bad. And you can see the idea of not having an adaptive immune system here in this green line, where you can see, okay, so the microbe comes in at the beginning, it reproduces up to this sort of max level, and then it's just there and never goes away, it just persists. You can't get rid of it, and it's potentially causing damage for a long period of time. And so that, of course, is one of many reasons why we know that the adaptive immune system is really important. But what I want to also tell you is I don't have a second picture here of a patient who is missing an innate immune system. Because we have actually never found anyone who is missing their entire innate immune system. Basically, not having an innate immune system is not compatible with being alive. You can't not have an innate immune system. It's just not possible. And so that should start to tell us, actually, it's probably pretty important. Like, the adaptive immune system, super important, but you can actually, be a human without one, even though there are a lot of issues there. It is not possible, it seems, to be a human without an innate immune system. We have never, in the history of the world, found anyone in that situation. We sort of hypothesize what would happen is what's shown here in the red line, is that if a microbe reproduced in that person, that microbe would just reproduce out of control and never be slowed down, never be held. It would basically just have exponential growth forever in that person. And you can see, it's taken off really early, it's going to a much higher level. And so what I want you to note from this is that while, if you look at the course overall, the percent of time we're going to spend talking about the innate immune system is way less than the amount of time we're going to talk about the adaptive immune system. It's not because the innate immune system isn't important. It's just the adaptive immune system in some ways is a little more complicated and takes a little more time to think through. So that speed difference that I told you about is just one part of this table, but it's a really, really big deal that people often underestimate. So there are sort of two other ways that I think about big differences between the innate and adaptive immune system. One of them has to do with how broad or general the recognition is. That gets into this thing that says limited number of specificities or numerous highly selective specificities. The idea is this system super, super, super picky about exactly what it's detecting, or is it sort of detecting something broad? So for example, the adaptive immune system can tell the difference between each of the variants of COVID. And this year's influenza virus, and last year's influenza virus, and next year's influenza virus. It is super, super, super specific. Specific is like a key word we love to use with adaptive immunity, how specific it is. For some reason, everyone uses the phrase, the exquisite specificity of the adaptive immune system. It's so specific. The innate immune system, by contrast, recognizes things more generally. The innate immune system cannot tell the difference between this year's flu and last year's flu. It actually just says, oh, that's an RNA virus. And any RNA virus, whether it's flu or coronavirus or measles or anything, triggers basically a very similar innate immune response. The innate immune response is just kind of like, that's a foreign thing. That's a different thing. I don't know exactly what it is. I'm not going to focus on exactly what it is, but it's something that shouldn't be around. And so it's very broad in its recognition, not very general. That is good for us as an organism, because if when we think about really specific responses in adaptive immune responses, we could imagine that a microbe could mutate and avoid the adaptive response really easily. Sort of like the adaptive immune system is so picky in what it sees, if you wore those little fake glasses, it wouldn't recognize you anymore. A little change, and it can't recognize you anymore. It's too specific. While the innate immune system is generally recognizing things that are broader and are things that a microbe can't change about itself very easily. An RNA virus can't stop being an RNA virus really easily. The other part to that is that if you were to count how many different receptors the innate immune system needs, you get a low number. I'm making up a number because we don't have like an actual number, but it doesn't really matter for our purposes. Let's say the number I make up is 100. There's 100 receptors, we'll say, in the innate immune system. You know, an RNA virus one, and a DNA virus one, and a gram-negative bacteria one. Make a list like that. So I said 100. This is also 2. In the adaptive immune system, in order to get the specificity that you see here, we actually have 10 to the 16th different receptors. A little bigger. And so that's one reason why some people get really excited about the adaptive immune system, is there has to be a lot more variability in the receptors. There has to be a lot more kinds of receptors than works in the innate immune system. One of the other reasons why this is interesting is if we look at the human genome and the number of genes, because again, these are, spoiler alert, these receptors are proteins. And proteins are encoded by genes, you know, basic central dogma stuff. The genome is 2 times 10, is that right? Yeah, that's right. 2 times 10 to the 4th genes. So somehow we're going to be able to get 10 to the 16th different immune receptors when we don't have nearly that many genes. And so the adaptive immune system has to use fancy genetics in order to make those numbers work. And so that's one of the other parts about the innate or the adaptive immune system that's cool, is that, you know, we end up with all of this variation, all that's giving us all of this specificity. It involves some of this fancy genetics. It involves all of these really fancy, really specific receptors. While the innate immune system kind of works like proteins that you learn in biology, and sort of, you know, pretty straightforward and pretty general. The other thing that is really different is that the innate immune response doesn't change over time. The adaptive immune response does change over time. It, in fact, adapts. That's kind of how it's got the name, adaptive. Why we changed the name to adaptive. And so the innate immune system doesn't change over time, so that's another way you could say it's sort of fixed in terms of constant during the response, while the adaptive response improves during the course of the response. It varies. Specifically, another way of phrasing that is that the adaptive response, when it gets better, it remembers your past infections. and improves based on that. So it shows a property known as memory in the adaptive immune response. You can see that example here. So on the left, again, we've got the innate immune response in the lighter blue and the adaptive immune response in the darker blue. And you can see the first time you encounter some microbe here. Here is the innate response. Here's the adaptive response, taking its slow time to make a response. But then you can see what happens if you were to be infected a second time with that same thing. The innate response looks identical to how it looked before. It does not change. While the adaptive response looks super different. In fact, you don't really have a delay anymore. Now the two are working together, you have a bigger peak, you actually have different slopes, there's all sorts of things that are different. And this is sort of this memory property. So the adaptive immune system adapts, while the innate immune system stays the same over and over and over again. And so again, that's one of the reasons why people get all hyped up about adaptive immune responses over time. This is a key part of how the adaptive immune system does all of the things that it does. I could spend so much time talking about this. I'm not going to right now, we're going to see more of it later. But it's a principle that is so important in immunology and in thinking about the adaptive immune system. I want to sort of get it into your brain now so that we can be hearing it over and over throughout the semester. And it in fact involves one of the biggest misconceptions people have about the immune system. So this is related to the adaptive immune system. And specifically, you can see here that it's related to that diversity, that 10 to the 16th business. Those different receptors, those 10 to the 16 receptors, shown here as 7 different colors because it would be really hard to draw 10 to the 16 different things. The receptors are all slightly different shapes and the cells are different colors. That is actually generated when the cells are first developed, not when you're getting infected. That's actually, you actually develop that full breadth, that full diversity, basically when you're born. You already got them all, but you only have like one of each. We're going to say one. People have actually done a measurement of what the numbers are. I don't want to get into that now, but we're going to say one for now. You got one of every single one of those 10 to the 16ths right now. You have one for Ebola right now. You have one for some pandemic virus that's going to happen in 2050. You already got the one already. You have one of every single possible one already right now today. And those are going to float around your body. Most of them are going to float around your body their whole life and never find their microbe and die. In fact, some of my colleagues talk about this as they die of unrequited love. They are basically going to go forever and never find their thing. I sincerely hope for all of you that your Ebola cells go for your entire life looking for Ebola and never find it. But if they do find their match, their microbial match, then that cell divides a lot and makes basically a clone army called clonal selection. That clone is selected. And now that you have a clone army instead of just one of that cell, you can start to have some activity and some actions. That's part of how the adapting happens, is that you picked that clone and made a lot more of it. And so this sort of diversity process that happens early happens before any pathogen. You already have a response to every microbe that you're ever going to respond to. Or at least you already have a cell with the receptor for every microbe you're ever going to respond to in your whole life, but you only have one. And it could potentially adapt by making a sort of a clone army of itself. And this is going to be the key underlying principle of all of adaptive immunity, is this clonal selection process. So that whole big mess that we talked about, can be divided into barrier versus innate versus adaptive. We can also divide it in some other ways. And this quote that I showed you last time from Mark Davis kind of gets at some of that as well. Because we can also divide this up into things like, are we talking about the cells? Are we talking about the molecules? Are we talking about the organs? And so, because we don't have kind of like the one set place that is your immune system, we kind of have to think about dividing up the immune system in terms of the cells, or the molecules, or the organs as well. I have this slide only for the purpose of introducing one term, which I kind of hate, but the field uses it, like everyone else loves it. I just have like a personal vendetta against it. And so I feel like you're gonna read it, so I gotta tell you what it means. And it's related to what we're talking about now, but I'm probably not gonna say a ton about it. And that is actually this term of humoral immunity, or humoral immune response. The idea of the humoral immune response goes back to a debate that was happening like in 1900 between early immunologists, one of whom Ellie Metchnikoff is from here. And they were basically, they knew that the blood had something to do with immunity. At that point, see, blood. They knew the blood was important in some way. And they knew the blood had cells and liquid in it. Liquid actually has proteins inside, and the molecules inside. But basically what they knew was there was cells and there was liquid. And they debated, are the cells the important part, or is the liquid the important part? And basically that's do we care about the cells or do we care about the molecules? When we think about responses that really involve a lot of work by the cells, we call it cellular immunity. We think about responses that really involve a lot of work by certain molecules, we call it humoral immunity. And that's because that was in the liquid, and they went back to like the 4 humors of like it being that like one of those 4 humors of ancient medicine of being off. And so people, basically, if we're going to talk about sort of molecules being important, sometimes we talk about that as a humoral response. And if we're going to talk about the cells doing a lot, we're going to talk about the cellular response. I spent so much time as an undergrad being confused about that. And the reason I was confused about it is I was like, don't the molecules come from cells? How do we separate the molecules and the cells? Where else the molecules come from? And that's totally true, and that's why still to this day I don't love this distinction. But really it's about, is the molecule the thing that's doing the important job against the microbe, or is it the cell? And there are some ways that immunologists still distinguish things as humoral versus cellular responses. If you look at the course schedule, you'll see that on Friday I'm talking about innate immunity humoral, and then next week I'm talking about innate immunity cellular. So like it can be useful as a distinction sometimes. It's not a distinction I'm going to spend a lot of time with. Really what I want to spend some time with now is thinking about the cells of the immune system. So we're going to start with thinking about the cells. This paper came out in 2023. I don't know if I recall if I mentioned, I co-host 2 podcasts, one This Week in Biology, one Immune. We covered this paper on podcast, which is how I learned about it, and it's fabulous. It actually tries to count how many immune cells are in a human body. And it does all sorts of really interesting stuff. And so in some ways we're going to be thinking about this. Note that they counted how many immune cells are in the human body in 2023. Also, what I want to point out is they had a 73 kilogram reference man. And so there can be some variation here. But when I'm telling you about the cells in the human body, in fact, you have 1.8 times 10 to the 12th cells. Yes, if you're doing math with my other math, if you're doing some calculations right now, it doesn't work. That's a later problem. So lots of cells. And one of the things that they broke down in this paper was actually where exactly all of the cells of the immune system lived in different organs. I'm going to come back to this and say more about this later on today. But for right now, I want to point out one key piece of this, which is that when immunologists, especially old-timey immunologists like Ellie Metchnikoff, I showed you on the previous slide, when they were starting to figure all this business out, they always looked at the blood, because it's pretty easy to get blood and start figuring this out. And so for a long time, they were like, the immune system is in the blood. We're going to care about the cells in the blood. It's all about the blood. And in fact, much of what we've done to like list and name immune cells is by looking at the immune cells in the blood. The blood is shown up here, and the blood is actually like not necessarily the majority of the immune cells. So again, in the past 10 years maybe, we've realized like, oh yeah, blood isn't always the best answer. But so much of the field has been done based on the blood. I'm going to be telling you mostly about cells in the blood as I tell you about cells. But know that like the blood might not be perfectly representative of the whole body. And you can see it is a pretty small fraction based on what they're seeing here. But it's what everybody does. And so it's how everybody talks about these cells. And so we're really going to be thinking about cells in the blood. This particular topic is one where I am the most sad that I don't still have the lab section attached with this course, because the first lab involves actually me showing you blood under a microscope and looking at all of these cells. And so if you want to see that, I have tons of blood cells and microscopes, and I'm happy to let you see that. Because you'll see a lot of the things that I'm going to be pointing out here. So if you actually look at blood, you've got a bunch of different types of cells that you'll find. One of those types of cells are the red blood cells, which are also known as erythrocytes. That's why blood is red. Erythrocytes, classically at least, don't really have an immune function. I've seen a couple that are kind of cool more recently, but we'll say we actually as immunologists generally throw them away. We don't care about them. One thing that is actually cool about red blood cells that will come up later is that red blood cells are unique in that red blood cells don't have a nucleus. So the only cell of your body that does not have a nucleus. They actually lose their nucleus during development. So red blood cells have no nucleus. And so immunologists often just think of them as like these trash things that carry iron. There are some entities called platelets, which come from a type of cell called a megakaryocyte, really important in blood clotting. Immunologists also generally don't care. But you can see both of these things really easily under a microscope. Again, for other reasons we probably should care, but I'm not going to get all into, but classically we don't care. The thing we really care about are a subset of cells in the blood called the leukocytes. And the leukocytes are white blood cells. If you look at blood under a microscope, mostly all you see are red blood cells, and you'll see like one little white blood cell here and there. They're way more rare. And leukocyte is a broad term that means all white blood cells. But we can actually divide up those white blood cells into groups. So there's one group that's called the granulocytes, there's one group that's called the monocytes, and there's one group that's called the lymphocytes. So those are the three big groups of leukocytes. Students often will confuse the terms leukocytes and lymphocytes. Leukocytes mean all white blood cells. Lymphocytes are a group within leukocytes. And you can also see, and this is actually true for all of the groups, but there are more cell types within them. So there are 3, for example, major types of granulocytes that you can see here. This shows, oh, sorry, Jazz.
Sorry, can I
Yep.
back that last slide?
Yep.
The
Oops.
slide
Yep.
previously regards like the separation or concentration of where immune cells are. I saw bone marrow was like a huge proponent and had like a large composition of immune cells because of its relation to like hematopoiesis and things like
Yep.
that. Or, okay.
Yeah, a lot of it is that. But there are a few different reasons, and we'll get to some of that. But so we'll definitely spend some time with bone marrow, and a lot of it is for those reasons you mentioned. So this is just, again, if we're looking at blood, and we're looking, again, in a milliliter. So I want you to think for a second about a milliliter of blood. In a milliliter of blood, you have 5 times 10 to the 6. That's 5 million red blood cells in 1 milliliter of blood. You have some number of platelets. I think about platelets so rarely, I don't really think about it. And you can see you have about like 3,000 in terms of some of your white blood cells. So the white blood cells, the total number of white blood cells is 7.3 times 10 to the third, so 7,300. So it's way fewer than the 5 million red blood cells that are in that milliliter. So the white blood cells are way rare. Again, that's why blood's red, not white. And different types of cells that we're going to see in terms of our white blood cells are present at different numbers. So some of them are kind of the frequent ones, the ones you find a lot. Some of them are really rare. If we look just at the white blood cells, you can see the majority of them are a cell type called neutrophil. Then there's sort of a medium number that are lymphocytes, sort of a smaller number that are monocytes, really low numbers that are things like basophils. And so the numbers are different. It's not like we have an equal proportion of all of these cell types. One of the other things that is super different about all of these different cell types is how long they live. So some of them live like a small amount, like days. Some of them live like years. And so that starts to impact both kind of functions they can have, what kind of responses they can participate in. That also influences kind of what their percentage is going to be like. So we've got lots of places where there's variation between all of these cell types. Jazz already sort of mentioned this, but one thing to note about all of the cell types in the blood, whether we're talking about red blood cells or white blood cells, and whether we're talking about the immune cells I've mentioned to you, or some that are even more rare, that will come up in the race here, it turns out all of those cells develop from the same original stem cell. So there's one kind of stem cell that basically makes all of blood. All red blood cells and all of your immune system. So all of it comes from this one type of stem cell. And we kind of understand this lineage of how all of these different cell types develop. The term for this process of the development of blood cells is called hematopoiesis. Hemata is the Greek word for blood, so not a shocker there. I actually didn't I've known for a while since I've done immunology for a while, but the Greek word that poiesis comes from is actually the same Greek root that is used for the word poetry, and it's to make. And so it's making blood cells, it's hematopoiesis. Immunologists, for some reason, we love to throw poiesis on the end of stuff. We make different things, it's the whatever poiesis. And if you're gonna write something about hematopoiesis, the number of things I have read in my life that have been like, we're now going to write about the poetry of blood cells. I'm a little over it. Like, it was funny the first time, but not so sure after that. So we can see this process of hematopoiesis where every single cell in the blood originally developed from this one stem cell, the hematopoietic stem cell. And you can see their sort of branching points. So the hematopoietic stem cell kind of decides between these two branches at the top, and then we've got these different like lineages that the cell can kind of go down. The hematopoietic stem cell, this is something I talk about more a little bit later, but the hematopoietic stem cell, it does come out of the bone marrow. This is a big part of your bone marrow. This is why people do bone marrow transplant. If someone does a bone marrow transplant, the point is actually that they're trying to transplant hematopoietic stem cells. That's actually what they're caring about when they do bone marrow transplants, are the HSCs. And you can see these different branches come up during hematopoiesis, and I'm going to mention, you're going to see these branches coming up a few different ways. So one branch is this sort of branch in the middle, okay? This branch over here. This branch includes red blood cells, who I've already told you I don't care about, right? I'm ignoring red blood cells and platelets, though they do come from this branch. And you can see this branch is called this thing called the myeloid lineage. And for some reason, people often miss that term myeloid. And when I use it sometime place later, they're like, the who? The what? Where is? I've never seen that word before. But that's the word, myeloid. It's also going to be on the top of the next slide. And that myeloid lineage includes the granulocyte group and the monocyte group of our leukocytes. So what you can see is sort of that hematopoiesis thing kind of works with this other way of breaking down these cells. So these things are not totally separate from one another. The myeloid cells, the granulocytes and the monocytes, also have another really important thing in common for your purposes. That is that the myeloid cells are the ones that do the innate immune system. And so if the hematopoietic stem cell comes down this branch of development and becomes any one of these kind of cells, it's doing innate immune stuff. Also, as I've said, I think that that many sentences I've said today in my brain have had a little asterisk in them. And so yes, there are exceptions to like everything, but for right now, myeloid cells, innate immune response. And so what you can see is that you've got this cell that sort of decides it's going to become a myeloid cell, and it can become that granulocyte group or that monocyte group. And all of those are our innate cells. One of those two groups that I mentioned to you is a group called the granulocytes. In this slide, and many of the slides to come, you are going to see images of these cells with this sort of pink and purple. If you come by the lab and want to look at any of the cells under a microscope, they will be stained with that same staining type. This is a super famous type of staining in the immune system. Like, immunologists look at those and like know immediately what they are. They're so classic that they do things like have necklaces. So you can wear a necklace of your favorite cell type. Or have earrings, so that you can have earrings of every single cell I tell you about, I have earrings of. So like it's a very famous sort of way of dividing these up. The granulocytes were named, and the people kind of made them a group because they all had some similarities under a microscope. And now we know, oh wait, they're related to each other. And they're all myeloid. And like they're all innate. Like, that was useful that they all looked the same. And the thing that they all had in common when you looked at them under a microscope was that they all had granules in their cytoplasm. They all had like these little circles, like little packets of stuff in their cytoplasm. We sometimes refer to them as being really granular. Sort of imagine them as little bags with lots of little pockets in them, lots of little packets. And so they got named granulocytes because they have a lot of granules. There are three main types of granulocytes that are found in the blood. Those are the neutrophil, the eosinophil, and the basophil. The big difference that we're going to care about right now is that they have different stuff in their granules, so they look a little different under the microscope. But in every case, they've got all these granules, all these dots. And they are all doing some kind of function in the innate immune response. Let me check something. Okay. So there's one other thing that is actually kind of interesting about them. This is most clear, and it's a thing that most people talk about when they talk about neutrophils. And if you look at the neutrophil up at the top, you can see you've got a lot of pink, and then these sort of purpley blobs. The purple with this stain, in most cases, is the DNA, is the nucleus, while the pink is the cytoplasm. My oversimplification for the moment. And so what you can see is that is not what you think of a nucleus looking like. That's this weird little blobby nucleus. It's called a lobed nucleus or a polymorphonucleus. Sometimes neutrophils are called polymorphonuclear cells because they have this weirdly shaped nucleus. You can go up to like 4 little blobs. The shapes are different from different neutrophils. I have like an image I took under a microscope of a neutrophil that looks like a smiley face. Like they have all sorts of different shapes. Honestly, until really recently, I don't remember what year it was, I just thought that was a thing. I actually didn't know that was important to how neutrophils work. It's actually important to how neutrophils work. One of the things that neutrophils do is they squeeze around to different parts of the body, and it's easier for them to squeeze if their nucleus isn't one big thing. It's easier to compress and squeeze around different parts of your body if your nucleus is like that instead of one big circle. So like and I just learned neutrophils have weird blobby nuclei, and that's how you pick them out under a microscope. But that actually is really super important in their being able to function. For our granulocytes, the big picture way that they work is they've got these granules, these packets, full of stuff. The stuff is different depending on which granulocyte they are, which is why they look a little different, how pink or purple they are under a microscope. And those are little packets of things that can kill microbes. And they might shoot them outside of the cell, they might shoot them at the microbe inside the cell, but it's basically little packets of stuff that they're going to kill microbes with, is how this is working. So we've got our 3 different granulocytes here, the neutrophil, the eosinophil, and the basophil. And you can notice that the neutrophil is by far the most common. There are a lot of neutrophils. Neutrophils are the white blood cells that you have the most of. Definitely in your blood. You can see that eosinophils are pretty rare and basophils are super rare. There's a lot that we actually don't know about basophils. And part of the reason why we don't know about them is because they're so few, they're hard to find. When we look at them under the microscope in the lab, sometimes students don't see one during lab period because they're so rare and so hard to find. And I'm like, that's okay, because it means the donor who gave this blood got to keep their one basophil in their body. They're really, really, really rare. One of the things that we're actually realizing more and more, so I have a former student who was in this class, and I remember his face when he was hearing this, and he was like intrigued with basophils. He now does basophil research and is like super into basophils. And one of the things that he would tell us, and that many immunologists would tell us, is actually part of the problem might be that basophils just live in places other than the blood. So we might have a bit more of them. We might be underestimating them because we spend so much time thinking about the blood. So you can see sort of these different cell types. There are also some other types of granulocytes that really are not in the blood, that are in other tissues, but they are still granulocytes in that they have granules. And so I want to at least mention them and introduce them now. Yep, Jazz.
In more tropical regions of the world, is it more common for them to have a greater percentage of basophils, considering like the implications of parasites in the water?
They usually will have more eosinophils, actually. So the eosinophilia is more pronounced. So one thing that sort of Jazz implies here is that the number of some of these white blood cells can change in different situations. One thing also to notice, especially I notice this when I look at the neutrophil data here, is what the range is. That is the range in a normal human. Like, and we actually don't really have a great answer of whether 41 or 74 is better. We have actually done a pretty bad job of defining some of these things and like what normal should look like. But there are times when your numbers will change dramatically when you, instead of having, 7,000 white blood cells, you'll have 20,000 white blood cells or something like that. You'll have a high white blood cell count. The reason why you generally have a high white blood cell count is because of your neutrophil number changing. So neutrophils, I insult neutrophils a lot. And I'm not going to insult them today. I will leave my insults for next week. But there are actually a lot of neutrophils in the bone marrow, and if you get an infection, the bone marrow releases them all to go deal with it, and the amount of white blood cells in the blood changes because you release them all into the blood, out of the bone marrow. And so if you go to the doctor and you have a blood count done, and they say you have an elevated white count, that means you have an infection. That just means that you have released some neutrophils out of the bone marrow. And in fact, this blood count, this sort of blood count is a key feature that's done in a lot of whenever you have blood work done, you will be able to get your complete blood count, which is basically just this information. So as I mentioned, there are other granulocytes that exist, though they are granulocytes that tend to be found in tissues and not in the blood, and so I will mention those after I answer Natalie's question.
Hi. So there's like this new little trend that people are eating like whipped bone marrow.
Mm-hmm.
Is it like debunk it? But if you want to, but like, is it like kind of like you can kind of obtain more neutrophils by eating it, or is that
No,
just like
because they're gonna go into your stomach and get digested by the stomach acid.
Okay, because that's what I kind of thought, but it's just like such a trend where they're giving like their kids like whipped bone marrow because they think it's gonna help.
I mean, bone marrow is also like a delicacy in some places.
Yeah.
You can imagine maybe it has like particular proteins that when you digest it will help your bone marrow a lot or something, like I guess, but like no, you're digesting those cells, you're not getting more neutrophils. The neutrophils are not like living through your stomach, going into your intestine, living through your intestine, leaving your intestine, like no.
It's just such a big trend, so I just wanted to ask you that.
Yeah, no.
Okay. It's just one of those things, I guess.
So the major granulocyte that lives in tissues and not in the blood is called a mast cell. And so you can see the mast cell up here at the top with a sort of that same staining. So you can see lots and lots of granules. You can also see a mast cell with a different type of microscopy stain here. So we've got the nucleus and we've got all of these little granules. The mast cell, like I said, lives in tissue, so you've got mast cells in your skin, you've got mast cells in your lung. And if something happens to those mast cells, the mast cell basically shoots all the granules outside of it to try to kill stuff. So this is a mast cell that's lost all its granules because it's tried to kill something. Those mast cells are particularly important for dealing with parasites. They are also the key mediators of most of your allergies. So I insult mast cells a lot, but they deserve it because of what they did to me. There is debate in the field. So I would say from my reading that I say, well, mast cells and basophils are super related. Like mast cells might just be like grown-up basophils or something kind of thing. My former student who works on basophils told me that was false, that they're not as closely related as I thought they were. But they are still all granulocytes. He does know a lot more about basophils than I do, so okay, fine. But the point here is that this is a granulocyte, but it's not really found in blood. The other really important myeloid cells are these cells called monocytes and macrophages. Technically, monocytes and macrophages are different types of cells. The monocyte can grow up and become a macrophage. For our purposes, I'm mostly going to use these terms interchangeably. technically the monocyte is the version in the blood, and it grows up in a tissue to become a macrophage. But again, like I said, I'm going to use them pretty similar. I'm going to use the terms pretty interchangeably. They have this sort of kidney bean shaped nucleus. That's really easy to see. They're not always, perfectly circularly round. If I ever, by mistake, am writing on the board, there's a way that immunologists like to often abbreviate macrophage. And so if I'm writing, you write this and forget to define it. Macrophage is usually M and then the Greek letter phi. So if I write M phi, I mean a macrophage. So we often will see monocytes in the blood, maybe macrophages, since we're not going to distinguish between them at this point. Sure. These are sort of, you can see in sort of like a middle-ish range in terms of numbers of cells in the blood. They're myeloid cells, so they are innate immune cells. And there's also another cell type that is sort of related here called the dendritic cell. Dendritic cells also are sort of part of that whole monocyte macrophage group. They also might be another way a monocyte grows up. They are not found in the blood. And the big reason why they're not found in the blood is this is a nice picture of one. I have actually a better picture of one on a poster in my office. They have all sorts of these little projections coming off of them. In fact, it's really important for them to have a lot of surface area. They're really big and stretchy. If that went through the blood, with the force that's actually going on cells in the blood, those would all get torn off. Blood cells pretty much have to be perfectly round to not get destroyed. That would get destroyed super fast in the blood. But in tissues, it's going to be great, and it's going to have lots of surface area to reach into all sorts of nooks and crannies. It is related to monocytes and macrophages. I'm not going to say a ton about dendritic cells, actually, until we get to adaptive immunity, and that might be weird, because dendritic cells are myeloid cells. They are cells of the innate immune system, 100%. But a key part of what dendritic cells do is dendritic cells help turn the adaptive immune system on. And so there's not a ton of their unique functions I can talk about until I'm actually talking about adaptive immunity and what needs to go on to turn the adaptive immune system on. And so so here's our hematopoietic stem cell in brown. Here we can see the branch of going towards the myeloid cell, and then it's not showing us the rest of that branch. The alternative choice is it could become something called a lymphoid cell. And if it becomes a lymphoid cell, it makes cell types that are called lymphocytes from the lymphoid cell. And the lymphoid cells are the adaptive immune system. And so this is sort of a nice sort of distinction. So we've got our lymphoid cells as part of the adaptive immune system. What I want you to notice in this image of the lymphoid cells is that how many branches there are, how many different kinds of lymphoid cells there are. I swear, sometimes I look at my immunology colleagues and they're like, I discovered a new lymphoid cell that responds to fungi in your left pinky on Tuesdays. I'm like, okay guys, we don't need to get that specific. So for right now, for today, until we really get into the adaptive immune system, they're lymphocytes. And we don't have to distinguish them. We will spend plenty of time distinguishing them later. Right now, they're just, the adaptive immune system cells are the lymphocytes. So yes, you can see some of them here. You can see the sort of classic lymphocyte, this nice big nucleus with a little bit of cytoplasm around it. There can be slight differences as we start to look at the different types of lymphocytes. Different types of lymphocytes do different kinds of things. We'll care about that when we talk about the adaptive immune system. Right now, we just need to know lymphocytes are the adaptive immune cells. And do all the fun adaptive immune stuff. And you can also see that lymphocytes are probably about our second most frequent cells in the blood after neutrophils. And this is, again, just throwing them all together as lymphocytes. So this is adaptive, this is all innate. So you can see there's probably way more innate cells than adaptive in your blood. But we're going to spend a lot more time talking about these adaptive cells because they have a lot of bells and whistles. This is actually something I also didn't know until I actually learned this from this paper, and I felt really dumb when I learned it. But this is actually telling you like the total number of all of these different cell types in the human body. So if you really want to know how many of them are in your body, it's listed here. At least if you are a 73 kilogram reference man. But also this tells you how much they weigh. So in total, all of these cells, if you are a 73 kilogram reference man, they weigh 1.2 kilograms. And what I didn't know, I had never thought about before, is that they don't all weigh the same amount. Some of them are humongous and some of them are tiny. And I literally never thought about that. And in particular, the macrophages are really heavy. And I work on macrophages, and I didn't know that till I read this paper, of how they're like the super giant cells. So this is actually showing you at the top, like percentages based on number of cells. You can see it's, a lot of neutrophils, a fair amount of lymphocytes. This is actually if you do the percents by how much of your weight they are, and you're all macrophages, at least in terms of your immune system weight. I literally did not know that till I read this paper, and I was just floored. So the other thing that we need to think about, which I'm only going to be able to really kind of briefly introduce you to today, and I'll have to finish up on Friday, is sort of organs in the immune system. So where are all of these cells? Where are they? What are they doing? Again, you can see there are lots and lots of organs that have immune cells, including, as it shows here, others that aren't even the main ones have a lot. They also showed this plot that they made where they looked at how dense that we have immune cells and how big the organ is. And so some organs are like big organs, but they don't have many immune cells. And some organs are like little, but they're all immune cells. So it sort of gets tricky in terms of defining all of this. But we as immunologists think about certain organs as the organs of the immune system. And in some ways, this goes into how many immune cells are there, but also what the cells are doing. And really the biggest distinctions I think of when I think of the groups I'm going to tell you about are what the cells are doing in each places. So we have 3 types of immune organs. So we're going to take all the organs of the immune system, all the places where these cells are, and divide them into 3 groups. We're super creative with our names. So one group is called primary, one group is called secondary, and one group is called tertiary. And so we're going to have 3 groups: primary, secondary, and tertiary lymphoid organs. And many key lymphoid organs are shown in this image. You can see we've got 2 of them shown in red. And the 2 organs that are shown in red are the bone marrow and the thymus. Those 2 are the primary lymphoid organs. There's only 2 primary lymphoid organs, bone marrow and thymus. So here you can see primary lymphoid organs, bone marrow and thymus. The bone marrow is the spongy tissue inside all of your blood. The thymus is this little tissue that some people don't know about that sits actually on top of your heart. It is not the thyroid, which is in your neck. We're talking about the thymus, which sits on top of the heart. When we get to talking about a lot of details about the thymus, I will tell you about how it changes over time, how many physicians think it's not important, but it is. How, in fact, many physicians actually cut it out if they do heart surgery and it's in their way. But we'll talk more about it. But it is one of the 2 primary lymphoid organs. Also, if I'm being technical, because I was having a conversation with that same former student recently, technically these are the primary lymphoid organs in humans. And there are some In some other animals, there are different ones. So we're not going to talk about that very much, but I was telling this other student that there were different ones, and he was like, felt like he'd been lied to. Like blown away when I was like, no, that's not how it works all the time. Also, I mentioned earlier that bone marrow is a delicacy. Thymus is also a delicacy in certain situations, particularly cow thymus. If you've ever heard of something called sweetbreads, that's cow thymus. When I had it once, it tasted salty. Like, I don't have anything grand to say about it, but that is a key thing. So the thing that's important about the primary lymphoid organs is what is happening there. So the cells are doing a specific thing in the primary lymphoid organs. And the primary lymphoid organs is where we see development of all of our red blood cells. So this is where the hematopoietic stem cell is. This is where the hematopoietic stem cell develops into all the other types. This is where kind of the cells are doing all of that fun genetics to make all those receptors before they get released out into the rest of the world. So developing your blood cells is where it happens in the primary lymphoid organ. They don't actually like try to kill pathogens here. They just grow up here. Sometimes people talk about, the thymus as T cell college. I actually have a book that calls the Thymus Murder University, and we'll talk more about that when we talk about the thymus as well. But the idea here is that this is where the cells are kind of educated, or where they develop. Then we've got the secondary lymphoid organs, and there are a lot of secondary lymphoid organs. They're shown here in yellow. So you can see we've got lymph nodes, you can see we've got spleen, but we also have the appendix and the tonsils and the adenoids. We've also got some specialized tissue in the GI tract. There are a lot of different secondary lymphoid organs. When most immunologists think about secondary lymphoid organs, we think about the spleen and the lymph nodes. So generally, I'm going to say spleen or lymph nodes. If I talk to you at some point about, like, if there's a problem and I'm like, and blah, blah, blah happens in spleen, or blah, blah, blah is happening in lymph nodes, I want you to kind of be thinking secondary lymphoid organ. Like, if I mean a secondary lymphoid organ, I'm going to say spleen or lymph nodes. They're the ones everybody studies, though there are a bunch of other ones that are used for different situations. And on Friday, we're going to start talking about how secondary lymphoid organs work. Because we need a little backstory before I can tell you what happened there. So that's where we'll start on Friday, and I will see you guys then.